Okay, so good afternoon, everyone. Okay, so today we'll be continue the important genera of Trichostrongyles nematodes in Abomasum. Yesterday we covered Homunculus, barbel poleworm, important parasite of sheep and goat, and also common in cattle in USA and also in different part of the world, especially in the tropical and subtropical regions, now emerging in the temperate regions. So today will be covered three most important parasites: Osteotagia. These are the brown stomach worm, one of the most important parasite of cattle in USA, especially in the temperate zones, and also the Tildosagia, a similar worm, brown stomach worm, but this is the Osteotagia of sheep and goat in the cold and wet climate. Trichostrongyles xi is a hairworm, is a little bit of vast host like in ruminant. It reside into the abomasum, and in the case of pig and horses, they reside in the stomach. Okay, so let's start with Osteotagia. Osteotagia has only one single species, that is called Osteotagia. Osteotagii cause infection in cattle. Very important parasite of adult cattle, normally causes severe disease in adult cattle. Worldwide distribution. This parasite is normally present in the cold, wet climates. In the more temperate region, is not a parasite of tropical part of the world. After the introduction of this microcyclic lactone, ivermectin type of drug, the prevalence of this species goes down because the Osteotagia is more sensitive to this drug, and resistance is not yet reported, and but there is a very few cases of resistance has been reported last few years. So parasite of Abomasum, small reddish brown in color. So this parasite you need to be very focused if you want to see that one in Abomasum. It will because it's less than 1 centimeter long, but in the case of Homunculus, that is 2 to 3 centimeter long, much easier, reddish color, easy to locate in the Abomasum in the necropsy. These eggs are brown, dark brown blastomeres. So again, you will not be say this is Osteotagia eggs. This is the Trichostrongyles eggs if you look it into the microscope. Prepatent period is 2 weeks. Okay, so who gonna explain me the life cycle of Osteotagii? Any volunteer? Mm-hmm. So just step back. If egg in the feces, then larval 1. Within the feces, within the eggshell, yes, and then L3. Once the L2 come out from the feces in the posture, converted into L3. So what is the main characteristics of L3? Infective, 1. Second, cuticle. They have a strong cuticle on the skin that protect that larvae from the environment, and then this L3 stage larvae goes into ruminant, and in this case is the abomasum, and before that juvenile L4 and L5 stage larvae, and L4 is the stage if they go into the hypobiotic stage, and then finally adult emerge. So this is how the life cycle of these Trichostrongyle parasites look like. Okay, so you have your point solution ready. You can do some knowledge questions. Okay, so which of the following stage of larvae is present in the cattle feces on the posture? Okay, so yes, L1 and L3, L2 stage larvae in the feces, and then they come out and converted into L3 stage larvae. Okay, so seasonal transmission of the Ostrategia. As I said, this is the parasite of cold and warm temperate region. So if this parasite is in the warm temperate climate, where the grazing is all year around, so highest transmission you will normally see in the between winter to springtime, right? And the larvae go into the hypobiotic state at the end of the spring, and then survive in the abomasal glands during the hot summer time. So it means that parasite know in the late spring that the hot summer is going to come in the warm temperate climate, and I have to go into the hypobiotic stage. On the other hand, in the cold temperate climate, where the grazing are seasonal because it's much colder, like north of Canada or north of Sweden, so the transmission is normally happen in between summer to fall, and the larvae goes into the hypobiotic state in the late fall, and because they will be survive in the cold winter in the host. So this is how the difference between the warm temperate as well as cold temperate climate as far as the seasonal transmission of this parasite is concerned. So hypobiotic state, who gonna explain what is hypobiotic state? Anyone? Yes. So they will be stay, and which stage they will be stay? L4. So L4 stage is the stage where they will be going to the hypobiotic state and to survive from the harsh or adverse environmental conditions. And normally it takes 3 to 4 months, but sometimes it depends on the outside environment. They can move up and down. So the warm temperate region like southern USA or southern part of England, these L4 will be arrest over the summertime. So this is what I explained in the previous one, what is the difference between the warm and cold temperate region. So in the case of cold temperate region like north of USA or Europe or Canada, these L4 will be go into the arrest state over the winter time in the host. Okay, so based on the previous information, now we can a little bit talk about the epidemiology of this group of parasite, where there's a two type of disease, clinical type 1 disease or clinical type 2 disease. What happened in the clinical type 1 disease? That is called summer osteogiosis. The posture larvae contamination is very rapid in the midsummer, and because the temperature become increased, and then they reach into the L3 stage larvae in the late summer. That is happening in the environment, in the grass, in the posture. So this L3 stage larvae ingested by a large number of L3 that goes into the host and cause the clinical type 1 disease. That is happen in the summertime and is very common. On the other hand, what is going to happen in the winter osteogiosis? Maybe some of the larvae they will be go into the hypobiotic state in that particular time, and these larvae become arrest into L4 stage larvae in the abomasal gland. Right? Once they become arrested there, they will be stay there. And the disease in that case is occur in late winter when this L4 stage larvae complete their development based on the external environment and then cause the disease called clinical type 2 disease. And that disease is normally seen and called winter osteogiosis. Right? Okay, so based on this one, which stage of larvae goes into the hypobiotic stage in the definitive host? Okay, 4. So, as far as the pathogenesis of this disease is concerned, the larvae will disrupt the abomasal mucosa and then also destroy the gastric glands, as shown here in this picture. In the postmortem, you will see the bacterial overgrowth, or the pH is high, and the putrid smell you will see. Mucosa look like a nodular appearance, edematous and hyperemic. So here you can see the L4 stage larvae just within these gastric glands. So here you can see the abomasal lesions. You can differentiate between the normal and abnormal abomasal mucosa. In the normal, very smooth, pale, but if you look into this abnormal one, you will see the edematous and roughy abomasal mucosa. So here you will see the edema. So that is seen here in the. Let me show you here. So these are the nodules. You can see they are raised as a nodule formation and edema. And this one is very important. You can see clear nodular appearance in the abomasal gland. Okay, so as far as the clinical feature of this disease is concerned, as I said, larvae is responsible for the disruption of abomasal gland, and the pH of the abomasum become raised due to the replacement of the HCl secretions that will be responsible for the watery diarrhea in this disease. So, what is the clinical feature of haemonchosis? Bottle jaw and anemia. So here you will see the watery diarrhea, but in the case of haemonchosis, you will not see the watery diarrhea. Right. And then the pepsins inhibit the digestions of the protein, and the albumin leakage through the abomasal mucosa that is responsible for the weight loss or reduced milk production and scar formation. So these are the other main sign you will see after in the clinical features of this disease, but watery diarrhea is the most characteristic sign you will see here. Okay, so you have seen the nodular appearance in the abomasal gland. Which of the following trichostrongyle disease caused in cattle? Okay, great. Osteotagia austragii. So, the treatment plan for osteogiosis, ivermectin, that belongs to microcyclolactone group, is the drug of choice, and this drug works perfectly in this case, and the resistance is not developed against this drug in this group of parasite, and that is the parasite is more sensitive to this drug of choice. So now we can start Osteotagia Tildosagia circumcenta. This is the parasite of sheep and goat. Both Osteotagia austragii and Tildosagia circumcenta, they belong to the same genus. The life cycle, biology are very similar. The only difference is the Tildosagia circumcenta is infect sheep and goat, and Osteotagia austragii infect cattle. And previously its name was Osteotagia circumcenta, is the parasite of sheep and goat. So this parasite is very highly pathogenic in sheep in all over the world, and especially in the temperate regions. Is also common in New Zealand, Scotland, Australia, Canada. This parasite is present, but not very much important in USA, but still in the northern part of the United States, the prevalence of Tildosagia circumcenta is very common, and in Canada is highly prevalent parasite there. So, important parasite of small ruminant sheep and goat. Abomasum is the site where this parasite reside. Very similar to Osteotagia, less than 1 centimeter hair-shaped parasite, and the eggs appear to be a blastomere, and we called it Trichostrongyles egg. So, who going to tell me that how many worms are enough to cause the clinical disease in Haemonchus? 500. So in this case, it's not very much pathogenic as compared to Haemonchus. Is 500,000 or more egg cause a clinical disease. Pre-patent period is 2 weeks, and how it will be look like. The L3 stay larvae that will be infect the host infect the host in the first 2 days, and then they might migrate into the abomasal mucosa. You can see these larvae in the abomasal mucosa, and from here they cause the disruption of abomasal mucosa, and then finally adult emerge and they come out into the surface of the abomasal mucosa. So now you can understand that life cycle is really important if you try to understand the, let's say, this pre-patent period, how this pre-patent period work, and how the L3 stage larvae to further convert it into adult, and what is going to happen in the abomasum. Okay, so now someone else will be now explain to me the life cycle of this parasite. Now we talked about the life cycle of these parasite couple of times. Who gonna explain? Any volunteer?
It's the same as.
Yes. So just explain. I mean, yeah, the same.
Adults shed the eggs into the feces. L1 develops in the feces, then turns to L2 in the feces, and then L3 comes out of the feces, comes to the pasture or wherever, which is then, I'm assuming, also covered by a cuticle protection. Then it gets ingested by the definitive host, either one of the small ruminants, which then enter into the abomasum.
Great. Yep. Okay, so the clinical features of this disease, we called it tildosagiosis, hyperplastic inflammation of the abomasal gland, that is responsible for the loss of weight and appetite, and hyperproteinemia due to the leakage across the mucosa, and you will see the animal is very weak and loss of weight. So Micro-leather appearance of the abomasal mucosa will be seen here in this picture, and the impaired abomasal function that is responsible for the pH of alteration in the pH, and that is cause the watery diarrhea and loss of appetite. So again, because it's very similar to Osteotegii, watery diarrhea is the most clinical feature in this disease. So, treatment plan for tildosagioses. Benzimidazole, ivermectin are the drug of choice. However, resistance to benzimidazole is one of the most important or threatening problem in this group of parasite. On the other hand, in the case of Osteotegia, the resistance is not the problem at this stage. So we developed this resistome technology, that is the high throughput technology with the high throughput sequencing, and if you will send me the samples and want to know that in this particular form, what is the resistance status? Shall I use this drug? Shall I use some other drug? Shall I use the combination? At least I will be tell you this is the resistance status in this form at a minimum level of 5%. So that is the sensitivity of this technology. So we just develop it and then applied into the field situation. Now we are looking forward to be further do some diagnostic application, take it into the diagnostic application of this technology. That is something to be worked with the commercial companies to be used as a diagnostic tool. Okay, so control strategies. The control program is always depend on the knowledge you will have how this infection built up in the host. The farming situation is very important. Pasture contamination, we talked about the pasture contamination, that which stage, L3 stage larvae cause pasture contamination. Seasonal appearance, we talked about that one too. The refugia management or targeted selective treatment plan. So we will be talk about these two plans, this management plan in our control of Trichostrongyles lecture. And the diagnostic test. So we will be perform the diagnostic test fecal count in the next lab. And this is one of the other technology I would like to share with you. This is called Nemobiome. My PhD supervisor, he developed this technology in Calgary, and I'm using in a number of different projects. And again, this is the technology. Now he already commercialized that one, by the way, in Canada, that farmers send the sample and they will tell them what particular species in these feces. Not just like these are Trichostrongyles eggs. So they will tell you that these particular species are in your sample in this form, and what now then you have to decide what will be the treatment plan of this particular form. So that's the new technologies now emerging, and they play a very important role for the accurate treatments of these parasites. Okay, so which of the following is the pathogenic nematode parasite of sheep in a cold climate? Case Tildosagia circumcenta, Osteotegii is the. Okay, so let's start with this one. Which of the following is the pathogenic nematode parasite of cattle in the cold climate? Okay, Ostrategia. Okay, so here we will be start Trichostrongyles xi is a stomach hairworm, and this parasite has a broader host range like cattle, sheep, goat, horse, pigs, and in the case of ruminant like cattle and sheep and goat, this parasite reside in the abomasum. In the case of horse and pig, this parasite reside in the stomach. So this parasite is also very tiny, like Ostrategia and Tillosagia, less than 1 centimeter hair-like worm. It's very difficult. You can see that one is the smallest worm of this Trichostrongyle family. So on the necropsy, maybe you will not able to see this parasite in abomasum. So the features of this parasite, like for example, like biology, epidemiology, and clinical features, usually is not a primary pathogen by itself. So previous the one we talked about in our previous part of the lecture, most of the parasite we talked about start from Haemonchus to Tillosagia. They are a single species infection. They cause a very severe infection themselves. But this case, Trichostrongyles xi is not a primary pathogen. It make a PGE complex. Anyone remember what is mean by PGE? Anyone remember that? Parasitic gastroenteritis complex. So more than two parasites, they can cause infection together. So normally you will be see this parasite maybe sometime with the Haemonchus placei, because in cattle they also make a PGE complex, and you will see that one in Haemonchus placei. Less pathogenic as compared to Haemonchus, Ostrategia, or Tillosagia, and they cross infect in a mixed species grazing situation. That fairly unique characteristics of this parasite is not a severe pathogenic parasite at all. Okay, so what we have covered in the abomasal parasites yesterday's and today's lecture. We covered Haemonchus, highly pathogenic, blood-sucking parasite, Ostrategiae, Tillosagia circumcenta, highly pathogenic parasites of cattle and sheep and goat, and Trichostrongyles xi, is not a pathogenic but make a PGE complex. So let's start with the summary of Haemonchus. What we learned in our lecture, both species, Haemonchus contortus and placei, is common in the warm temperature or climate, most favorable. The parasite is more favorable for this tropical or subtropical part of the world. It causes very severe disease in the case of sheep and goat, blood-sucking parasite. In the case of cattle, it is not as severe as compared to sheep and goat. The largest parasite, abomasal nematode, 2 to 3 centimeter long. Barbal pole appearance, we covered it. It causes severe anemia and submandibular edema. That is the main characteristics of this parasite. Is the primary pathology due to the adult worm. So the adult worm cause the disease. How? Adult worm cause disease. How they cause this disease? What is the clinical features? Sorry? Sorry? Anemia. So what they're gonna do? The adult. This bloodsucker. Yes. So they cause, because they're sucking blood, so that's why adult worm cause disease. Tropical parasite, but one of the emerging parasite in the temperate regions. Ostrogii is the important pathogen of cattle, very small. Hypobiotic is the main feature of the transmission of this of disease, and go into the hypobiotic state, that is L4 stage larvae. But in this case, L3 stage larvae is most pathogenic stage. Why?
Whenever they're active, like during the L4, they're like staying away from the harsh climates.
Yes, you are close. Anyone want to add something? Anyone? Why they are pathogenic? Larval stage is pathogenic. So we talked about Haemonchus, adult stage is pathogenic because they suck the blood. In this case, the larval stage is pathogenic because they destroy the gastric glands. Right. So, cause type 1 and type 2 disease, summer ostrogiosis, winter ostrogiosis, and the most important parasite in the cold climate, and the transmission occur in the USA in the cold season. Tildosagia circumcenta, the only difference is this parasite is cause infection in sheep and goat. Ostrogii cause infection in cattle. Trichostrongyles xi, most common parasite found in cattle and sheep, but also have a broad host range. They infect horses and pigs, and a very small parasite is not a primary pathogen, may contribute into a PGE complex with some other parasite, and this parasite cause disease together. Okay, so anyone has any question? Any question? No? Okay, great. Thank you.